Hello and welcome to a brand new video on the muscle contraction cycle. So in this video, what we're going to discuss is the organization of a whole muscle. So we're going to look at from, say, for example, biceps brachii and break it down to its smallest parts. Then we're going to look at the structure of a muscle fiber that's going to be useful for us to have an understanding of. The basic structure of the sarcomere, and I do mean basic. We're not going to be looking at a detail composition of sarcomere here. That will be in another video. The structure of both thick and thin filaments, and then how the cycle for contraction and relaxation of a muscle. So what we want to do is start ourselves and build up a whole muscle. Now, to build up a whole muscle, what we want to do is start off and say that we had a whole muscle. So that might be, for example, the biceps brachii, the rectus femoris muscle. And then we will see that whole muscles are made up of individual fascicles. And the individual fascicles are made up of muscle fibers. A muscle fiber would be found in here. Muscle fibers will be found there. And muscle fibers contain myofibrils. And these myofibrils are made up of smaller units called sarcomeres, the sarcomeres. And lastly, your myofilaments. Myofilaments. That will be the thick and the thin filaments found inside of a muscle cell. Thick and thin filaments. Now, what we want to look at here is, as we understand that whole muscle is made up of individual fascicles. Fascicles are made up of muscle fibers. Myofibrils are made of sarcomeres, and sarcomeres are made of myofilaments, which are the thick and thin filaments found in a muscle cell. Let's now get an idea of the muscle fiber. Now, just as a quick recap for the basic anatomy of a muscle cell, and you can see this in my very first video posted to my channel, was for the neuromuscular junction. That the cell membrane of a muscle fiber is the sarcolemma. The sarcolemma has these little openings in it called a transverse tubule or T-tubule. And the T-tubules, remember, are joined on each side by bulges of smooth endoplasmic reticulum called the sarcoplasmic reticulum. These bulges are called the terminal cisternae. And one T-tubule and two terminal cisternae makes a triad, because 1 + 2 = 3, triad. And that the terminal cisternae hold a lot of calcium to regulate muscle contractions. And that's the important thing, is that that's concentrating. Now, inside we could see our myofibrils here. And if I were to pull that myofibril out, I am going to be able to see the sarcomeres. So let's take and just give a very quick introduction to the sarcomere. Now, the sarcomere is made up of thick and thin filaments. So the thick I'm going to be drawing in blue here, and we're going to make them a little thicker, of course, than our thin filaments. So this will be a thick filament. And thick is made up of mostly myosin. Now, thin filaments, let's use red to do our thin filaments, and let's draw this considerably more thin here. And we're going to label these as thin. And thin is made up mostly of the protein actin. Myosin is thick, actin is thin, and together these are called myofilaments. Little ditty that sometimes I use, but I do put a lot more emphasis on that when I'm in class. Now, what we want to see is one of the most important things I can really begin to outline here for today is this region here where the thick and the thin filaments overlap, called the zone of overlap. Now, why is zone of overlap so important? This is the area in which the thick and thin filaments will interact to cause a muscle contraction. Now, how do they do it? How is that going to be possible? Since we started with thick, let's look at the structure of thick filament. Now, diagrammatically, I have that over here. Now, I used blue for thick filament. So, thick filaments have two major parts to it. They have a fibrous tail area, and then they have a globular head. Now, the heads we can think of as an ATP ACE because they have ATP binding sites on them that will bind to this protein, highly charging it and be able to power muscle contraction. The tail is an extremely fibrous component that allows for it to be bundled, and bundles of this will come together to make a thick filament that we can see with all the myosin heads kind of sprouting off of that, which will be able to bind to the thin filaments. Now, the thin filaments themselves, as we look at them here, since we drew them in red, they are made up of the protein actin, and actin here, we have what is called a G-actin. G-actin, or globular actin, is each individual bead. When you take a string of these and then another string of these, you have made F-actin, or filamentous actin. That is two strands of G-actins. And they're held together by a protein called nebulin, which acts as a glue. Now, besides G-actin being present, now the G-actin here is going to have what is called an active site, or myosin binding site, and it is regulated by another protein here, and this protein is called tropomyosin. Tropomyosin is the protein that one of the major proteins that regulates the myosin-actin interactions. Along with that is that protein here called troponin. Now, troponin has three individual subunits that I'm going to talk about later on in this video, but troponin and tropomyosin regulate actin and myosin's interactions to regulate a muscle contraction. So, as we go through this, I'm going to do a very simplified version of what we're doing. And how I'm going to discuss it throughout the video here is since these are thin filaments, let's draw these in red. And we're going to start out in my video. I'm going to draw 1, 2, 3, 4 of these always when I draw it, just for simplicity. And these are my G-actins. Now, we're just going to draw one strand of the two, but this is going to be our F-actin. So you have, remember, your G-actins here. And then tropomyosin, we're going, troponin tropomyosin complex. So tropomyosin, and then troponin and its three subunits. So we have G-actin. We have tropomyosin. We have the three troponin subunits. And when troponin and tropomyosin come together, we have the troponin, tropomyosin complex. And these regulate the actin and myosin interactions. So when myosin is here, wants to be able to interact with it, it can't, because tropomyosin will be covering the active sites, the myosin binding sites, okay? So now we mentioned there are actually 3 subunits of troponin who do different things, and they form a complex. A little bit more of a realistic impression of this would be something more like this, where we have our, over here, if we had our G-actin in its active site, there is one molecule of troponin here called troponin 1. Then there is this molecule here in the middle called troponin C, who has calcium binding sites, 1, 2, 3, 4 of them. And then there is this guy here called troponin T, because he binds to tropomyosin. And tropomyosin and troponin form a complex that cover these active sites of G-actin, okay, and prevents myosin from binding. Now, let's kind of take an analogy for this and help us understand how this all works and how troponin C and the ions called calcium cations can bind to this and affect a muscle contraction. And it is very important that we understand calcium's role in this. So I have kind of an analogy I want to use to do this. And I'm going to start out like this was a date. Now, you can try to draw this with me a little bit, step by step, but we're going to think about this like it's a date. And in this date, there is a young man, Myosin, who really would like to go out and date this young lady named Actin. Now, Actin here, and Myosin wants to hold hands with Actin, but Actin will not be able to. And why is that going to be? Is her father, the troponin-tropomyosin complex, does not want Myosin to hold hands with his daughter, G-actin. So what is going to happen here is when they come in there to ask her out and go take her on a date, and Daddy is sitting there being like a typical father might do, is saying things like, what is your intentions towards my daughter? You better have her back by 7 o'clock. Things like that. And in America, it's very common for fathers to kind of threaten the first boyfriends a little bit. So Mama Calcium, though, she was in the kitchen here, Mama Calcium, and she was in the kitchen, Ca++. That's the terminal cisternae. She's in the kitchen, she sees all this happening. And so what happens is when Mama Calcium comes out of the kitchen, she's going to ask Daddy, hey, I need your help with something. Can you help me open this jar of snacks for our guest? And so he moves out of the way here because Mama Calcium here is bound to him, and Mama Calcium is got him distracted a little bit. So he's now out of the way. So that means now this active site has become exposed, and so that means Myosin can now go over and hold hands with G-actin. He can bind to our active site. He can grab her by the hand and hold hands with her now. But once Mama Calcium goes back into the terminal cisternae, back into the kitchen, and she's no longer here distracting Daddy, what's going to happen is Dad is going to come back and he's going to end up not wanting these two to hold hands anymore, and he is going to get back to covering up her active site, and Myosin can no longer hold hands with daughter Actin. So now we're going to be back to a relaxed state here. So kind of think about it is, daddy is troponin-tropomyosin complex. Mama calcium comes out to distract daddy so that myosin can hold hands with G-actin during their date. So this is basically what's going to happen. So now let's go through a step-by-step kind of process for this as we look at every single step along the way of this and discuss it. Now, what I want to begin myself is we're going to start with a relaxed muscle fiber. So we're going to start here with our young man here, this boy, Myosin, who wants to hold hands with the little girl, G-actin. Now, he is going to be highly energized. When a young man might be going on a date, he's going to be full of nervous energy, very nervous about his date. And it's ATP. Let me erase that and start over. I'm going to use a different color here just so I don't get anybody confused with my drawings. So what we want to do here is have our myosin head highly charged with ATP. So he is in a high energy conformation. He is full of nervous energy. And the first person that he is going to meet for his date, when he knocks on the door to ask out little Myosin, was daddy, troponin-tropomyosin complex. Remember, there are 3 myosin troponin units on that. And there's going to be 1, 2, 3, 4. Let's draw 4. Let's draw a 5th one here. Doesn't really matter. I'll erase that because I'm trying to keep it to 4 just for simplicity for my drawings. But the active sites here are covered up by your troponin-tropomyosin complex. So, but he is in a high energy conformation. Now, what is going to happen here is that in the very next step, as we look at this, is this ATP energy is bound to that. Now, remember, during our date here, we had our thick filament myosin, this young man, who was full of nervous energy here at his date, very nervous about it. And we know that daddy was the one covering up the active site. But mama calcium comes out. And when this calcium level goes up, what is going to occur is that will bind to the troponin subunits, to troponin C. And what is going to happen is your 1, 2, 3, 4. Let's draw some of these here. Let me erase some. I want to keep it to 4 just for simplicity. And sometimes it's easier said than done when I'm drawing this out free form. Is this tropomyosin complex, where we had 1, 2, 3 troponins, is that this calcium binds to one of them. And basically what happened is in this unit that we drew here, that we drew in class, we drew together here, is that calcium, there were these calcium binding sites, 4 of them, and calcium starts to bind to this, and this is going to move out of the way. And it's going to take a conformation more like this. And the active sites are exposed here. The active sites are exposed. So when calcium binds, troponin-tropomyosin complex rotates out of the way here, exposing the active sites. So the very next thing we saw was the active site exposure. So this is what we can say here is active site exposure. Now that the active site is exposed, now that Mama Calcium has come out of the kitchen and has distracted Daddy, the very next thing that's going to happen is Myosin, who's full of that high stress nervous energy here, is, because remember, he is full of nervous energy, he is going to come in here and bind to the active sites here. He is going to do what we call a cross bridge, because at this time the troponin and tropomyosin complexes have troponin C has calcium bind to it, and the active sites are exposed, and we call this linking a cross bridge. Now remember, this is full of energy. So what is going to happen here that's very interesting next is after cross bridge has occurred, you will then have this myosin head here who had the ATP bound to it and full of that nervous energy here. What is going to happen? Now, if you're drawing with me, just hold up for a second. You will see that energy will be used. This ATP was broken down into ADP and an inorganic phosphate. So it used that energy to pivot the part of the little rotating piece of it, the little flexible part of that protein. And when it was bound to one of these, what it has done is also move the thin filaments here. And when it has moved these thin filaments here, we saw that we moved this. We call this burning of ATP here to move the thin filament. We call this the power stroke. The power stroke. It is bound to it, cross bridge, then power stroke, burned the ATP. ATP energy was consumed. Now that ATP energy is being consumed, the power stroke has happened. We have to break the interactions between our thin filaments here, our G-actins, active sites. We have to break the bond that was there by our myosin, okay, our myosin heads. So what we're going to do is we're going to take our myosin here, and I am going to give it some ATP to break the cross bridge. Now, this is why rigor mortis happens. Rigor mortis occurs because we have consumed energy upon death. We're not making any more energy. We cannot produce ATP to break cross bridge. So what's going to happen is you will stay in rigor. But if you do have energy, you will break the cross bridge, disconnect this, and put it back to the high energy conformation. All right, and now that its high energy conformation is present, then what is going to happen, because basically he's been holding the daughter's hand and scared now, and Daddy's got him separated. What is going to happen here is that myosin, who is in his high energy conformation, is now going to be separated from the daughter again. He's not going to be able to hold hands with daughter actin, not going to be able to hold hands to her active site. And why this happens is the calciums let go and they go back in the terminal cisternae. When calcium levels begin to decrease, tropomyosin goes back to its, and sorry, I should have had this here, 1, 2, 3. I do apologize for my little miniature mistake there. As calcium left that, you will see this goes back to the relaxed conformation. We now say it's relaxed. We now can say that this cell is relaxed. Now, what's actually happening is on your myosin, remember he was like this. Sorry, he was like this. Sometimes when I'm trying to sketch, it's a little bit more difficult than I expect. It was like this. And then what's going to happen is you're going to go to a conformation like this. With the active sites covered back up because calcium is bound to this troponin C. With all the calcium on your troponin C here, it's going to go back to its relaxed state. And so this is why the decrease in calcium causes relaxation, and an increase in calcium causes contraction. So, to kind of recap the overall process, that we began our cells with a relaxed muscle fibers where troponin tropomyosin complex was covering the active sites of G-actin. ATP was bound to the myosin heads with a high energy conformation. Then calcium binds to troponin C subunit, exposing the active sites. Then cross bridge happens, and that because of the active sites being exposed, myosin heads bind to the active sites on G-actin. Then the ATP is broken down or hydrolyzed, causing a power stroke. Then ATP is expended to break the cross bridge and return myosin heads back to a high energy conformation. Then calcium goes back into the terminal cisternae, causing troponin tropomyosin complex to return to their original position, to blocking actin-myosin interaction and causing muscle to relax. So I hope you found this video helpful to understand the complex biochemistry of the muscle contraction cycle. Please let me know of other videos you would like to see me do in the future. I'm excited to do other topics. I love teaching these topics. But if you find this video helpful, please leave a comment, a like. Don't forget to subscribe. Let me know if my videos help you, and good luck in your future endeavors. Thank you so much.